Good day, and today I would like to present our research on preliminary statistical analysis of amino acid sequence embeddings of proteins. The authors are Krzysztof Fidelis from University of California, Davis, USA, Yaroslav Teplyi, and Mykhailo Luchkevych from Lviv Polytechnic National University. Understanding protein structure is crucial for bioinformatics, impacting areas like drug discovery, disease modeling, and structural biology. Recently, large language models have been utilized to predict the 2D structure of proteins from their amino acid sequences. These models use high-dimensional vectors, also called embeddings, to represent proteins internally. Our goal is to conduct a statistical analysis of these embeddings to identify patterns in their parameters like linearity, stationarity, probability distribution, ensuring the correctness of applying corresponding models and methods in the future. So why protein structure is so important? Proteins are large and complex molecules that play many critical roles in the body. They are made up of long chains of amino acids, which are the building blocks of proteins. Each protein has a unique sequence of amino acids which determine its specific structure and function. The structure of a protein is crucial because it directly affects its function. Proteins must fold into specific two-dimensional shapes to function correctly. Therefore, understanding protein structure is vital for various applications. Protein structure can be divided into multiple levels. The first being primary structure. This is the linear sequence of amino acids in the polypeptide chain, and this sequence is determined by the genetic code. The secondary structure refers to local folded structures that are formed within a polypeptide chain. The most common types of secondary structure elements are alpha helices and beta sheets that consist of beta strands, which are then collected by the loops. The tertiary structure is the overall three-dimensional shape of a single polypeptide chain, and the tertiary structure determines the protein's functionality. So understanding the structural components of proteins is crucial for predicting and analyzing protein function. One of the key methods to achieve this understanding is through multiple sequence alignment. Multiple sequence alignment is a method used to align protein sequences to identify regions of similarity. These similarities can provide insights into structural, functional, and evolutionary relationships between the sequences. In our study, MSA is used to establish a baseline for comparing the similarity and dissimilarity of protein pairs. In the recent years, several state-of-the-art models have emerged for predicting protein 3D structures. Notable examples include AlphaFold2 by DeepMind, released in 2020, which revolutionized the field of protein structure prediction. Later, Meta's ESM2 model used end-to-end transformer architecture without a need of evolutionary information and MSA samples, and proved that the structure can be predicted from the sequence itself without any additional information. So this is the reason why we chose this model for our research. A few words about ESM2 model. It is built on a transformer-based architecture and utilizes self-attention mechanisms to capture long-range dependencies within protein sequences. Most of the protein structure prediction models use two representations of protein sequence. The first one is sequence embeddings. These are high-dimensional vectors that represent individual amino acids within a protein sequence. Each amino acid is encoded with information about its context within the entire sequence, capturing local and global relationships. The second type of representation is pairwise embeddings. These embeddings capture the relationships between pairs of amino acids in the sequence, and they provide detailed information about the spatial interactions and potential contact points between different parts of the protein. In this research, we focused on sequence embeddings. In our research, we have analyzed two pairs of proteins, one similar and one dissimilar. So let's take a look at the embeddings themselves. First thing that we noticed immediately is that each sequence embedding extracted from ESM2 model has outlier values at the same indices, where at index 274 we have extremely high value, and at index 37 we have an extremely low value. So we have applied normalization to remove these outlier values so they don't skew our results. We remove these values by taking the average of two adjacent values. After the normalization, the embeddings are consistent and ranging on average between -100 to 100. The next step was to calculate the magnitude of embeddings and compare its distribution between protein pairs. As can be seen from the histograms, the embedding magnitude distribution is comparable between similar and dissimilar protein pairs, where the upper histogram is a distribution for similar proteins and the lower one is for dissimilar proteins. To assess the stationarity of the process, we calculated running mean over the embedding magnitudes and fitted a trend line, which appeared flat and almost parallel to x-axis, supporting stationarity of the process. Next, we attempted to evaluate a theoretical probability distribution of embedding dimension cuts and embeddings magnitudes. So the distribution of embeddings dimension cut at a specific index and the mean value over all dimensions follows a normal distribution, where magnitudes of embeddings follow a log-normal distribution. Also, an interesting thing can be seen for the dimension cut distribution, where the histogram for the similar protein pair, which is this one, is largely overlapped. But the distribution for dimension cut in the dissimilar pair has a shift, which signifies the difference between the two proteins at this dimension. After statistical analysis, we have developed a methodology for calculating protein similarity based on sequence embeddings. For this, we utilized autocorrelation function and correlation analysis. On the picture to the right, we can see that the rows in the matrix represent embeddings of each amino acid, and the columns represent dimension. The ESM2 model uses 1024 dimensional vectors to represent a single amino acid in the sequence. To start the analysis of this protein sequence, we need to transpose this matrix first. This is needed because we will calculate autocorrelation function across embedding dimensions for each amino acid rather than the embedding itself. The left picture visualizes a process of autocorrelation function calculation for two proteins that are being compared. We use sliding window to calculate an autocorrelation for each dimension. So, for example, by sliding a window across the first dimension, we will have a set of autocorrelation functions for this dimension that will also be represented as a matrix, where the number of columns is W, where the W is the size of a window, and the rows are n minus W, where n is the length of the sequence. So at the end, we have a three-dimensional matrix of size 1024 by n minus W by W. Then we take these two matrices and calculate Pearson correlation between each autocorrelation function for a given dimension. For example, as can be seen on the slide, we take the first autocorrelation function for the first dimension of protein A and calculate Pearson correlation with each autocorrelation function of protein B for this dimension. As a result, we end up with three-dimensional matrix of size 1024 by n minus W by m minus W, where n is the length of a protein A and n is the length of a protein B. The last step is to calculate the mean value of correlation scores for all dimensions. So as a result of our method, we have a matrix that should reflect similarity of two proteins, where on diagonal we should see whether there is any similarity in the sequence or not. On this slide, you can see on the right a visualization of correlation matrix between two similar proteins, and on the left an MSA alignment of their protein sequences. On our correlation matrix, we can see that there is a signal on the diagonal, which means that these proteins are similar, which also aligns with the MSA results. Let's take a look at the more complex alignment. Here, the one protein is significantly larger than the other, and as can be seen on the correlation matrix, the sequence similarity signal is shifted off the diagonal, which means that there is a gap between sequence parts that are similar. This can also be verified by looking at the MSA results of these sequences. So as can be seen, the autocorrelation method we propose is able to detect protein sequences similarity from the ESM2 model's embeddings. So to conclude, we have conducted a statistical analysis of amino acid sequence embeddings generated by ESM2 model. We determined embeddings dimensions, magnitudes, and probability distributions, and compared between similar and dissimilar protein pairs. Also, we have developed a correlation and autocorrelation-based method for evaluation of protein sequences similarity and verified the results with MSA. A future work includes the development of embedding-based alignment method using autocorrelation analysis to further improve the accuracy of protein similarity assessment, and further development of the existing methodology to calculate similarity score between proteins so that we can compare our method with other existing methods. Also, we plan to analyze the pairwise embeddings. Thank you for your attention.